Hello viewers, welcome to the e-learning platform of Dorong College. Myself Dr. Kishore Deka from Department of Botany, Dorong College, Tezpur. I am going to deliver a lecture on the topic Herbarium techniques and its significance, which is covered in the paper both HC 4036 of BSc 4th semester and both 3036 of MSc 3rd semester of Guwahati University. Herbarium is a kind of art. It is a type of preservation systems where we preserve plants, their parts, leaves for a long periods of time, and along with the preservation, it is also used to study the specific plants. So in this video lecture, we will discuss about the concept of herbarium, the steps of preservation of a good herbarium and its importance. First of all, let us discuss about the concept of herbarium. Herbarium is the storehouse of plant specimens. This is a preservation method of Hausser specimens for vascular plants, that is for pteridophytes, gymnosperms and angiosperms. Plant specimens are pressed, dried, preserved and mounted and arranged in a sequences of accepted system of classification for further references. The herbarium may also be regarded as a museum. The name herbarium is referred to one or more rooms or a building where a mounted and level herbarium sheets are stored systematically with a wide representation of plant species from various geographical regions. Herbarium is a simple piece of paper which held the dried and pressed plant parts. The sheet is made up of handmade thick paper in the size of 42 by 29 centimeter. It is a standard size. One herbarium label is attached generally to its lower right corner to record different data related to that particular specimen. Now, we will discuss about the history of herbarium. Actually, the concept of herbarium was first given by Luca Guini in the period between 1490 to 1556, a professor of botany of Italy who first dried plants under pressure and pasted those on papers as long-term records. His students spread this art throughout Europe. In around the year of 1700, for the first time, Stanford used the term herbarium for a collection of dry specimens of medicinal plants. However, in 18th century, Carolus Linnaeus started a new method in which he mounted his specimens on single sheets and started storing them flat horizontally, which is followed by almost all the museums and herbaria in the world. Now, tools or equipments of plant sample collection. For the proper collection of vascular plant material and processing of specimens leading to the final insertion to the herbarium cabinet, a number of tools or equipment are required. So in this slide, I have mentioned some important equipments or tools which are used during the period of plant sample collection. So first one is the plant pressed with blotting paper. A herbarium press is a lightweight portable one, preferably made up of wooden plates of 12 by 18 inch. It is basically used to press and dry plant specimens for preservation and scientific study. The second one is the field notebook. Field notes gives all the information about a plant that cannot be preserved with that specimen. So field notebook is used to record different field data during the time of collection of plants, like collection date, collector name, collection number, coordinates, elevation, associated species, key characters of the species, common names, etc. Third one is the tag. Tagging is important to record the collection number and date. So plane cutter. Plane cutter is important to cut the stems and hard shrub branches. The fifth one is the hand lens. For quick observation of morphological parts, a 10x hand lens is essential. Next one is the Vasculum or collection bag. Vasculum is the lightweight metallic chamber with a strap for hanging from the shoulder. Nowadays, vasculum is rarely used. Polythene bags are more popular as it is easy to use and can carry more specimens. Small bottles, envelopes, etc. is basically used to collect loose materials. Camera is essential for taking close-up and zoom picture of the collected specimens. GPA is uh, also important to record the precise location of the plant as well as study area. Altimeter is used to record the altitude of the collection site. So apart from equipments, some other materials also may be required during the field work like liquid preservatives, wax, paper, first aid box, etc. Method of preparation of a good herbarium. So there are 5 important steps to prepare a good herbarium. So first one is the collection of plant material. Second one is the pressing or drying of plant material. Third one is the sterilization or poisoning. Fourth one is the mounting on herbarium sheets. And fifth one is the labeling. Collection of plant material. This is one of the most important steps to prepare a good herbarium. So we have to always remember at the time of collection that plant material should be with reproductive units. So if possible, at least 5 specimens of plants, preferably in all stages of development, should be collected. If necessary, 2 or 3 trips to the same spot must be made to collect different stages of specimen. All the 5 specimens of the same plant are given the same field number. In case of grasses and other herbs, the whole plant including the underground part should be collected. If it is woody, a twig that can easily fit into the herbarium sheet can be collected. Plants with underground modified stem are to be preserved along with those specialized organs. Very rare specimens like orchids, insectivorous plants and endangered threatened plants must be collected sparingly. If the plant is dioecious, like the species of Cucurbitaceae, Ericaceae, and some species of Euphorbiaceae, then specimens should be collected from each sex. Ensure that specimens are free from insect infection, fungal infection, and any pathological symptoms because it creates problems to identify specimens. In case of fern, it should be collected with their basal portion because the shape of rhizome and hairs and scales on rhizome are important taxonomic characters. Fronds of fern without sorae are of little value. Sorae have traditionally provided the most important characters for fern classification. The arrangement, location, and shape of soras are used by botanists to identify their different variants. Succulent plants like cacti, euphorbia, and members of Crassulaceae present unusual difficulties in making herbarium specimens. Their thick succulent tissues take very long time to dry and so they require special attention. Hence, either the tissue should be killed by dipping in boiling water or excess of tissues removed by hollowing out the thick organs. Uh, Treating with alcohol or strong formalin can also kill the tissue. While recording the characters of succulents, especially the spiny succulents, details of shape, size, and the arrangement of spines and joints should be noted. For aquatic plants, just after collection of specimen is placed in a tray containing water and is spread out, then a safe plate with which paper or muslin cloth is inserted below the specimen and taken out. The paper or cloth is lifted slowly with both hands and placed between the dryers. So in case of aquatic plant, the bloaters may be changed in several times in a day. Before putting the specimens in the plant press, tagging is necessary with field number and date of collection. Pressing and drying. This is a crucial stage for herbarium seed preparation. If the field or study area is a remote place and the collectors are not returning to the main laboratory on the same day, then one small portable plant press is to be used for initial drying in the field and specimens are to be shifted to a larger and heavy plant press on return to the laboratory. Folded blotting papers or, in its absence, folded newspaper can be used as blotters. While placing the specimens on the blotters, it is essential to keep at least one leaf showing the lower surface. It is important as the lower surface of lamina bears many important characters. Roots and underground parts should be washed thoroughly before pressing to remove soil particles. Timing for changing bloatings is very much related to the nature of the plant specimen. If it is a soft aquatic plant, then for the first day, change the bloaters in every hour to avoid many damages to the specimen. However, for normal mesophytes, give changes every day for the first 3 to 4 days at the beginning and then gradually increase the duration until completing the drying. In this slide, figure A is the wrong way of pressing of a specimen and figure B indicates the right way of pressing of a material. Herbaceous specimens longer than 40 centimeter may be folded into V shape, N shape or M shape and then pressed. There are some exceptionally tall herbs. In that case, those plants are pressed in three parts. There is one from the basal portion, another from the middle part, and third one from the topmost part of the plant, which contain the reproductive unit of the plant. Poisoning of dried specimen is very important as those are prone to become food for many insects and microorganisms. So to save the plant specimens from these organisms, one or more chemicals are used. The most popular is the mercury chloride. Generally 4 to 6% solution of mercury chloride in rectified spirit is taken in a PVC or enamel coated tray, as mercury chloride is one highly corrosive chemical. Many herbaria now successfully use luryl pentachlorophenate. A 3.75% solution of luryl pentachlorophenate in white spirit is used. Bundles of specimens are dipped in it for few minutes and then again dried under pressure. This is much safer alternative. Mounting. After a specimen is pressed, dried and poisoned, it is affixed along with a label on a mounting sheet. So mounting is the process of affixing a dried pressed plant and its label to a sheet of heavy paper. This provides physical support that allows the specimen to be handled and stored with a minimum damage. The aim of mounting is that the specimen should be neatly and uniformly spread and fixed on the sheet, and all parts of the plant should be easily visible for study. All herbaria even today use temperature sensitive glue as adhesive for mounting. It is having the benefit of opening the specimen easily by heating below the herbarium sheet, and that could be refixed by cooling. In some herbaria, instead of glue, specimens are attached to the mound using mounting tape. Narrow tapes are used for delicate plant parts and the wider ones for the stems of different thickness. Sometimes some parts of specimens need to fix with the herbarium mount by stitching using needle and strong durable threads. Thick stem, fruits, seeds, etc. are to be fixed in such a manner. So the last stage of preparation of a herbarium is the labeling. So after the mounting of a specimen on the herbarium sheet, the pasting of herbarium labels is done on the sheets. The size and design of herbarium labels slightly vary according to the need. Size is about 8 by 12 centimeter. This includes field number, date of collection, place of collection, name of the plant, family, habitat, local name, some characteristics. Those will not be available from a dry specimen, etc. So herbarium level is affixed on the bottom right hand corner about 1 centimeter away from edges of the mounting sheet. So finally the herbarium sheets are ready to work and for inserting in herbarium cabinets. Bryophytes and lichens are not mounted on cardboard sheets like other plants. They are kept in folded envelopes. Now come to the point, important herbaria in the world. So these are the some important herbaria of the world which contain a maximum number of herbarium sheets, of which herbarium of Royal Botanic Garden Kew is the most largest one, and it can maintain about 7 million of herbarium sheets from different regions of world. Similarly, other important herbaria are British Museum of Natural History, London, Museum of Natural History, Paris, New York Botanic Garden Herbarium, Herbarium of Mysore Botanic Garden, Royal Botanic Garden, Edinburgh, etc. So these are the some important herbaria of India, of which the Central National Herbarium is the most oldest and largest one, which is situated at Howrah of West Bengal, and it maintains about 20 lakhs of specimens from different parts of world. Another important herbaria are the Forest Research Institute Dehradun, the National Botanic Garden, Blotter Herbarium, and different regional herbaria of BSI. Now, come to the point, function of herbaria. Plant specimens are permanently stored in herbaria and therefore they are the major sources of information about plants and vegetation. Preserved specimens of herbaria are used almost all kind of taxonomic research. A picture of all species of a genus or all the genera of a family may be gathered only in the herbarium. The classification of world's flora is based mainly on the herbarium material. List of the endangered species of any region may be prepared only by the herbarium specimens. Monograph of genera or families are prepared only by the herbarium specimen. Big herbaria provide training facilities to young students in herbarium practices. Herbarium provide the national plant wealth and provide scientific information to the public regarding the plants. So this is all about the herbarium techniques and its significance. I hope this video will be helpful for you all. Thank you.